Why blotting techniques is used? What are the different direction in the name of direction, whether it is eastern, western, southern, that is also there, northern. Every direction is made on or name of that. The scientist is also given name of that scientist name. Southern blot by scientist name southern. Not every time same direction, but blot techniques are useful for transferring DNA, RNA, and protein. As you know that in central dogma also DNA to mRNA, that is transcription, mRNA to protein, that is translation. Now these floating techniques are for transferring DNA, RNA, and protein into a carrier. Why? So they can be separated and used often follow the use of gel electrophoresis. The southern blot is useful for transferring DNA. Northern blot it is used for or it is used to detect RNA, and western blot for protein. But in certain books also eastern blot is. But it is used to detect a post translation modifications. Now another how southern blot techniques or blotting techniques picturize form, how it is transferred. For example, you having a genomic DNA, use a restricted endonucleases and separate it on various gel electrophoresis. After that, you got that band also, and the desired traits you are putting in this southern blotting. How the southern blotting? It require a nitrocellulose paper. That is, many people are telling nylon paper, but it is a nitrocellulose paper. Here is agarose gel. You know, blotting buffer is floated on that, and below is nitrocellulose paper. So sandwich they are making and putting and absorbing above one. Likewise, they are high molecular weight and low molecular weight. You know very well, those having a higher molecular weight that on upper side and lower molecular weight in below sides. So southern blotting techniques is doing like this, and their nitrocellulose they are also utilizing certain probes. These probes is taking a specific things to attach with. Finally, you will find under the exposure of X-ray, that detection is which content is come on that paper. Southern blot techniques for what? For detecting specific RNA separated by gel electrophoresis. And it is hybridized with the DNA probe or labeled probe, as I said, this 32P. Southern blot technique. Who has developed this southern blot technique? Name itself indicate EM Southern. Now, another, what is the applications of southern blotting techniques? You can read.
Southern blotting technique is used to detect DNA in given samples. DNA fingerprinting is an example of Southern blotting. Used for paternity testing, criminal identification, victim identification, to isolate and identify desired genes of interest. Used in restriction, fragment length, polymorphism.
Okay, thank you. Now, as you know that these are the application of southern blot techniques. If it is asked in the examination, you have to write like this. At least these point you have to write. So you are not aware of molecular marker techniques or marker assisted selection. That's what the word is uh, looking like that. Now you will identify afterward, and southern blot technique is used to detect, as I said, DNA detections. So where it is applied? It is applied in forensic science also. For example, you want to identify the specific gene or specific persons identify or criminal investigation is also applied for that. Another things, it is also useful for paternity testing, whether these cross between these plant and these plant, whether it is mother character governed or paternal character governed, or certain times whether it is male and female cross, the newborn is same male or female parents are different. So paternity testing is also possible with southern blot techniques. Victim identification, when we are sometimes what will happen in certain cases we do not find any clues about any criminal investigation. Then you have to identify the certain whether they have touched with the floors or their DNA is fall down on that area. They have smoking with birds. Cigarette butts is also used for extraction of DNA. So to isolate desired gene of interest, that is one of the application of Southern blot technique used in restriction fragment polymorphism. Then to identify mutation or gene rearrangement in the sequence of DNA. Mutation, whether the mutation is happening or in particular tissues or not, that can also identify. For example, in many cases, when we are doing a mutation, for example, EMS treatment is given to a certain flower seeds or flower crops. After growing, it will require a number of times or number of things is also happening. So to choose the which, if you grow in that and you find a mutation or gene arrangement in the flower color, then you can extract this gene is variate from this parent also. So used to diagnose disease also, that can also apply southern blotting techniques. Southern blotting technique is also used to identify infectious agent. Sometimes what will happen, they are double-stranded DNA, double-stranded RNA. Then another techniques that will, but you have to identify whether they have any genetic nucleic acid present in that or not. Infected persons or infected genes or infected. Now another technique is northern blot techniques. You read loudly.
Northern blotting. It is also called as northern hybridization, technique for detecting specific RNA separated by electrophoresis by hybridization to a labeled DNA probe. Northern blotting was developed by James Alvin and George Stark at Stanford University in 1979 and was named such by analogy to southern blotting.
Pictures that is given for layman persons, not we people, for understanding those who are not knowing what is northern bloating or how the bloating is happening. So these pictures are shown like this for primary student, not for you like PhD student. So it is called a northern hybridization. Now what? I am not using bloating techniques. I am using northern hybridization. This technique is for detecting the specific RNAs separated by the gel electrophoresis and hybridized to labeled DNA probe. Remember that I have not used RNA probes, DNA probe. Northern blotting, why this name is given to northern blotting? As I said in the previous slide that the southern blotting, because of that, his name is EM Southern. Now, in the consequent or same things is applying, but here scientist name is not northern. James Alvin and George Stark at Stanford University was named. Why? Because of analogy to southern blot. They are near to that. Same practices he is applying. Therefore, this name is given. Now, application of northern blotting. What is the application of northern blotting? As I said, to study the RNA or study the degradation of RNA, whether the RNA splicing is possible or not. You want to study about RNA splicing, that can also be possible. Whether the RNA life, half life, it can also study with that. Similarly, expression, generally gene expression. Main things, many people are why isolating mRNA. They are studying of gene expression at the level of mRNA, or we can say mRNA transcript. So a standard for these application northern blotting techniques are useful for study of gene expression in the level of mRNA. That you have to remember, or mugging also. Only one word, right? Now third, that is Western blotting. Same thing. Gel electrophoresis is also used. But in both cases, you will find agarose gel electrophoresis. And sometimes they are also used, same also, uh, acrylamide gel electrophoresis. It is called immunoblotting. As I said, northern hybridization, southern blotting. Now I am telling another word. It is called immunoblotting. It is developed by W. Neil Burnett. Burnett, this technique for detecting specific protein that is separated by polyacrylamide PAGE, PAGE, polyacrylamide gel electrophoresis, and it is also, as I said in previous slide, they are utilizing a DNA probe. Here you will find labeled antibodies. Remember this word, labeled antibodies. The picture is also seen here, or Western blotting techniques. You want to antigen samples, then it is separated on the particular proteins, and these protein is blotted, put into a transfer into a nitrocellular sheet with the blotting tanks, and then finally it's labeled with the antibodies. And you can see with the autoradiography, with developed fixed autoradiograph, whether this is antigen antibody reaction is happening in that or not. This is all about the application of Western blotting. I will complete it within 1 or 2 minutes. This is, or Western blotting techniques are generally used in most of the protein identification. So it is having a medical application as well as in we people also. For proteomic studies, we are utilizing that. So Eastern blot techniques, that is given by S. Thomas, and that detect the target, as I said, post-translation modifications of the proteins or distinguish them from standard far. So these are the differences, comparison between southern, northern, and western blotting techniques. So you have to remember this word: molecular detected, gel electrophoresis, gel pretreatment is required, blotting methods, and which probes are used, and detection systems. So you can write like easily. For example, in southern blot techniques, they are taking double stranded DNAs, whereas in northern, as I said, RNAs, and western, you will find a protein. So gel electrophoresis used for that in southern blot agrosgel, a formaldehyde agrosgel, or a northern blot, that is polyacrylamide gels. In the gel pretreatment requires in southern blot depurination, denaturation, and neutralization. Three processes are involved. DDN. Okay. In capillary transfers is found in the both southern and northern blot, whereas in case of electric transfer is useful for the Western blot techniques. So DNA probes is both in cDNA and cRNA, radioactive or non-radioactive probes are used for northern blot techniques, whereas same thing is DNA probes are used in the southern blots. Western blot require a primary antibodies for the as a probes, you can say. Now detection systems for that, generally the three instruments are used. One is chemiluminescence, another is autoradiography, and colorimetrics. So you will find in the all the cases you will find this comparison detection system for Western blot, Southern blot, and Northern blot techniques. So this is all about the blotting techniques, and you have to